Assalamualaikum warahmatullahi wabarakatuh and a very good day everyone. We are from Group 7 and today we will present to all of you about passive transport. First and foremost, passive transport is the movement of ions and molecules across the membrane from higher concentration to lower concentration. Or in other words, the substance moves down the concentration gradient. Furthermore, passive transport does not require any energy to move the substance. And there are a few examples of passive transports, such as water osmosis and reabsorption of nutrients by the human intestines.
Devyashini anak perempuan Shanmugam, matrix number 217627. So I'm going to explain the types of passive protein transport. There are two, which are channel proteins and carrier protein. Channel protein are passive transport only, but carrier proteins are active and passive transport. So let's see the channel protein. It serves as passageway for the molecules to pass through. They form hydrophilic pores that extend across the lipid bilayer. When the pores are open, they allow specific solids such as inorganic ions with appropriate size and charge to cross the membrane. The example of ions are sodium ion, potassium ion, chloride ion, and calcium ion. So this is a diagram showing the channel protein. So this solute will go through this pore. Channels that remain open to both interior and exterior of cell is referred as pores. So this is the pore. But for special case, aquaporin is a water channel. Aquaporin is a channel protein that facilitates transport of water between cells. So let's see the channel protein versus carrier protein. Channel protein transport ions only, but carrier proteins transport molecules. Channel protein solutes molecule diffuse through the pores. But for carrier protein, solid molecules are bound to one side and released from the other side. Channel protein only transport water-soluble molecules, but carrier protein transport both water-soluble and insoluble molecules. Channel protein have high transport rate, but carrier protein have low transport rate. That's all from me.
Now, let's know more about carrier protein in passive transport. Carrier protein, also called carriers, promises, or transporters, bind the specific solid to be transported and undergo a series of conformational changes to transfer the bound solid across the membrane. Carrier proteins contain a substance and in doing so triggers a change of its ownership, moving the bound molecule from the outside of the cell to its interior depending on the gradient. But when does the carrier protein change their shape? So protein can change shape when their hydrogen bonds are affected. Like channel protein, carrier protein are typically selective for one or a few substances, where it has a specific binding site for a single substance. It shows the properties of plasma membrane that is selective to some substances. There are three types of carrier mediated transport, which are unipart, sympart, and antipart. For unipart carriers, it transport only one kind of substrate, for example, glucose. While sympart carriers move two or more substrates in the same direction to cross the membrane, for example, glucose and sodium ion. While antipart carriers move substrate in opposite direction. The example for uses of carrier protein in passive transport is the transporter channel. PLAT4 is incredibly important in diabetes. PLAT4 is a glucose transporter found in fat and skeletal muscle. Insulin triggers PLAT4 to insert into the membranes of the cells, so the glucose can be taken in from the blood. Since this is a passive mechanism, the amount of sugar entering our cells is proportional to how much sugar we consume. Up to the point that all our channels are being used, it is saturated. In type 2 diabetes mellitus, cells do not respond as well to the presence of insulin, and so they insert PLAT4 into their membranes. This can lead to soaring blood glucose levels, which can cause heart disease, stroke, and kidney failure.
An ionophore is a substance that is able to transport particular ions across a lipid membrane in a cell. Structurally, an ionophore contains a hydrophilic center and a hydrophobic portion that interacts with the membrane. The net ion flow occurs only down an electrochemical gradient. There are two classes of ionophores, which is mobile ion carriers and channel formers. The example of carrier is valinomycin, which is a molecule that transports a single potassium cation. The valinomycin transports K plus down the electrochemical gradient by picking K plus on one side of the membrane, diffusing across the bilayer and releasing it on the other side. This carrier binds to a particular ion and shields its charge from the surrounding environment. This facilitates the ion's passage across the lipid membrane's hydrophobic interior. However, at very low temperature, these ionophores lose their ability to transport ions. The other one is ramicidine, which is a channel. The channel formers that create an opening in the membrane, which is hydrophilic, allow the ions to pass through without coming into contact with the hydrophobic core of the membrane. Two chromicidin molecules end-to-end cross the bilayer to form a transmembrane channel. It allows monovalent cations to flow down their electrochemical gradients. Ionophores that create channels are typically big proteins. Unlike the carrier ionophores, these forms of ionophores can continue to transmit ions at low temperature.
So now let's move on to the next part, which is voltage-gated cation channels. Voltage-gated cation channels are transmembrane protein that plays an important role in electrical signaling of cells. It is also important in propagation of action potential in neurons. What is action potential? Action potential is traveling waves of electrical excitation, or we call it as nerve impulse. And action potential is triggered by depolarization.
Okay, next, I will share about the depolarization of plasma membrane. In nerve cells, a stimulus that causes sufficient depolarization promptly causes voltage-gated sodium ion channels to open, allowing a small amount of sodium ion to enter the cell down its electrochemical gradient. The influx of sodium ion, which is positive charge, depolarize the membrane further, thereby opening more sodium ion channels, which emit more sodium ions, causing further depolarization. This process continues until the electrical potential in the membrane has shifted from its resting value of negative 70 millivolt, almost as for the sodium ion equilibrium potential of about positive 50 millivolt. At the equilibrium potential, the net electrochemical driving force for the flow of sodium ion is almost zero, and the cell will come to a near resting state with all its sodium ion channels permanently open. Sodium ion has an automatic inactivating mechanism which causes the channels to close rapidly even though the membrane is still depolarized. The sodium channel remain in this inactivated state and unable to reopen. Voltage-gated potassium ion channels open so that the transient influx of sodium ion is rapidly overwhelmed by an influx of potassium ion, which quickly drives the membrane back toward to the potassium ion equilibrium potential, causing hypolarization even before the inactivation of the sodium ion channel is complete. The membrane potential then returns to its initial negative value. If we look at the graph below, the green curve shows how the membrane potential would have simply relaxed back to the resting value after the initial depolarizing stimulus if there had been no voltage-gated ion channels in the membrane. This relatively slow return of the membrane potential to its initial value of negative 70 millivolt. Then the red curve shows the course of the action potential that has been explained before. The action potential is basically propagates along a myelinated axon by jumping from one node to another node of Granville, where the sodium ion channels are mostly concentrated at the nodes. That's all from me. Thank you.
Hi everyone, My name is Ramyashni Nanasegren and my metric number is 216829. Today I am going to talk about transmitted gated. So what is transmitted gated? Transmitted gated ion channel are specialized for rapidly converting extracellular chemical signals into electrical signals at the chemical synapse. So what is the process of transmitted gated? So the neuronal signals are transmitted from the cell to cell at the synapse, and the presynaptic cell is separated from the postsynaptic cell by a narrow synaptic cleft. So if we see the first diagram, the above is called as the presynaptic cell, and the below one we call as a postsynaptic cell, and the middle is known as the synaptic cleft. So the next is the change in the electrical potential in the presynaptic cell will trigger it to release a signaling molecule which is known as the neurotransmitter. So what will this neurotransmitter do? The neurotransmitter will be rapidly diffused across the synaptic cleft to provoke an electrical chain and will bind to the transmitted gated ion channel. As we can see in the second diagram, the neurotransmitter will be released by the synaptic vesicle through a process known as exocytosis, and which will be released and bind to the transmitted gated ion channel that is placed on the postsynaptic cell. So after the neurotransmitter has been secreted, it will be rapidly removed. There's a two-way that the neurotransmitter will be removed. One is will be removed by a specific enzyme. Second, it will be reuptake by the nerve terminal that has released it, which is the synaptic vesicle. So neurotransmitter is divided into two, which is inhibitory neurotransmitter and also exhibitory neurotransmitter. So inhibitory neurotransmitter, one of the example are like Y-aminobutyric acid, which is known as GABA, and also glycine. So the function of the inhibitory neurotransmitter is to regulate the brain activity to prevent problem in areas of anxiety, depression, sleeping, and concentration. So next is the exhibitory neurotransmitter. Example for the exhibitory neurotransmitter is acetylcholine, which is ACH, which is function in our central nervous system and also peripheral nervous system, where it will regulate our heart rate and also our blood pressure. So that's all from me. Thank you.